that laser pointer is everything you need?
Yep.
All right.
Thank you.
And I will be back down at 5 to
Okay.
collect everything.
It's like 1,000,000 degrees in here.
It's a good thing. It's pretty wonderful.
Yeah. Did you get my email before I sent like half an hour ago?
No.
Okay, well, you know where to come, I guess.
Yeah, yeah.
So that's good. Afterwards, for a few weeks, we've been having a little get-together of the colloquium speakers. Sometimes go to some of the younger faculty at Big Ed's for a beer.
Okay.
If you'd like to come.
Great. Yeah.
We'd like to have you come.
Sure.
That'll probably be the only dinner or lunch.
That's fine. Yeah.
I'm sure there wasn't too much expected, but
No. In-house.
yeah. But now let's see, what should I say for introduction?
Oh yeah.
So normally we do some of the history of
Yeah.
where you went to graduate school,
Okay.
postdoc.
So grad school was University of Chicago.
Okay.
Department of Chemistry. Molecular biology.
Biochemistry and molecular biology.
I wrote a thesis on cytochrome oxidase.
I think we just
Yeah, and then
probably just Chicago when.
right. That was in 19. I got the degree in 1995. So, and then I did my postdoc at UC Boulder.
Okay. And when did you come here?
2000.
Into biology department.
Yeah.
Okay. And you still do X-ray crystallography. So that's
Yeah.
When did you come to Chicago?
I started the degree in 88.
So we overlapped pretty much.
Oh, okay.
I didn't graduate from undergraduate there until I
All right. Yeah. Yeah. There was another undergraduate. Two courses together. Barb Golden.
the name, yeah, actually I did.
She's at now at
I don't know that I know her
Yeah.
well, but certainly I know the name, and I'm sure she's friends of mine.
And my daughter is now about to graduate from
Right, yeah.
It's a special place, especially for undergrads. For people like you and
Well,
Bart and my daughter.
yeah. Going back, because he had graduated last week, and when I went back, and the feel amongst the students there is so different than what I get from the feel of the students here. Or I was at Rutgers for a while.
It's very special.
And I tell people who think why should we pay for such a go
Yeah.
to
Yeah, there's a lot of people that understand it.
So the feel of the students are different, but also a lot of the instruction is pretty awful.
Yeah.
But that's not what you're paying for. You're paying for them to be with those other students. So
And the challenges, right? The challenges. I probably went over the story, but Minnie came. She had a really rough first year. She came on and did courses for a whole year.
So one issue.
It's 1 hour. So I planned for 1 hour, but
No, 1 hour is good. So one of the reasons we've been trying to have this session is to get more faculty to come. So I'm not sure if will come. The students have to come. These guys are students except for pro.
It's a course.
Yeah.
Just terrible.
I also now understand why adjunct appointments don't come more often in biology. This is a biology.
Very badly.
It's not something that we would realize. Just stick within your own way. Michael, are you going to come to the dance?
Yeah.
Great. Martin, I invited Martin.
Yeah, like I look at this, I'm like, yeah, that's kind of like
Molecular biochemistry and biophysics. Is that what the department?
Biochemistry and molecular biology.
Biochemistry and molecular biology, okay. I'm thinking at Yale there was a molecular biophysics and biochemistry.
Get Doug Gibbs. His office is in Yale. Call Seymour. He was at Chicago just before I got there.
Gibbs' office must have been in physics lab there somewhere, but I don't know that we knew where his office was.
Yeah, no, it's in that department.
I mean, oh, well, there's a new building. I don't know where they were.
This was when I was interviewing for the A. Whitney Fellowship. Must have been 96.
I don't know, the building was there, but but yeah, I always figured his office was in Slough.
It may have been.
Of course, I mean, I spent most of my time in the building named after him. As a graduate student.
it's like 1 hour full.
We usually start a couple minutes.
Yeah.
So, 4 o'clock, yeah. So.
I mean, it doesn't make sense.
It's a lot of construction going on. What are you guys doing?
I think that this construction is just building a new, like, heating, cooling.
Oh, okay, so it's all
But, so there was an old glue building there, but it's like twice as big in every dimension, so it's like 8 times bigger.
Yeah. And Physics 10, is that like one of the original buildings on campus?
The one next door here?
Yeah.
Yeah, it's been around for a while. I think it's old enough that they can't tear it down. Exactly. This building, we're hoping they tear down soon. That one, I don't think they can.
We have to, you have to make it, I think it's
It's the most convenient building I've ever. It looks nice. Everything could look nicer.
Life sciences.
Yeah, there's another building rooting for it to be torn down.
Well, I mean,
Well, I know.
yeah, but it's putting a big strain on space.
Right, well,
And a lot of faculty don't.
No.
Well, Tim,
They moved out of there, but very small. Sort of space, what's available.
we need to ask you. So,
It's a source of stress.
I'll add you to the WhatsApp. We should ask you. At least everything changes. Yeah, it probably won't matter, but you never know if they're going to be enforcing it or not. We better safe than sorry. It's like $4,000 fine for 6 months. It's great. You can't go there without the required. But for example, we went to no, without the change. Yeah. But they change. They love you. They change. I think they're only going to enforce it when the conditions are necessary. Yeah, right. Make sure that people have the proper. Don't need those in New York. I mean, they must have changed them. Or he's
Okay, I think
this though.
probably it's right. Okay, so today's colloquium speaker, in my attempt to bring in people from our university in different departments, it comes from the biology department. Martin Horvath. He got his PhD from Chicago in the Department of Biochemistry and Molecular Biology in 1995. And then he went to UC Boulder, where he was a postdoc, and then he came to Utah in 2000 and has been here since then. He studies protein structure using crystallography, and that's why I invited him, because it's kind of physicky to do the X-ray crystallography, and we'd like to hear about the structures as well. So,
Thank you. Thanks, Doug, for inviting me, and thank you for coming. So I put the title together without thinking as completely as I should have. Structural biology now encompasses many techniques, including cryo-electron microscopy, neutron diffraction, and we're just going to be sticking to something that I know things about, which is X-ray crystallography. And the outline of my talk is we're going to, for about half of the period, we'll be looking at a brief introduction to X-ray crystallography of macromolecules, and then we'll take a look at a couple of case studies of research work that in my lab in collaboration with people at UC Davis for the MUT-Y DNA repair enzyme, and with people in ophthalmology department up at the Moran Eye Center here in Utah, studying carotenoid-binding proteins. Okay, so the very beginning of macromolecular X-ray crystallography begins here with James Sumner, who showed that this enzyme, Jack bean urease, could be crystallized. And so this demonstrated a number of things which weren't known at the time. One is that enzymes have a defined structure, because only entities with defined structure can form crystals, and that this X-ray crystallography depends on the ability to grow crystals, and Sumner did this for Jack bean urease and then catalase, and since then many proteins have been crystallized since then in complicated forms. Even the ribosome has been crystallized and studied by this method. This is another iconic sort of advance in the field. Rosalind Franklin obtained this photograph of fibers of DNA, hydrated fibers of DNA. You can think of these as like two-dimensional or one-dimensional crystals. And then the diffraction pattern after X-rays are passed through that fiber gave clues as to the periodic helical structure of the double helix that was reported in this 1953 paper by Watson and Crick. Much later on, for a more complicated macromolecule of protein, myoglobin, Kendrew, using methodology worked out by Perutz, was able to obtain the structure of the first protein. And different from the DNA helix structure, the protein structure was this kind of visceral, ugly, grotesque kind of shape. There's no apparent symmetry, and so it was much more difficult to describe the overall structure by comparison with the very beautiful symmetrical repeating structure of DNA. In 1965, this is work by Phillips and people in Islam. They crystallized lysozyme, and this is the first enzyme. So a protein with catalytic properties able to catalyze chemical reaction. First enzyme to be determined using X-ray crystallography. Okay, so now going to sort of the methodology and where we find some physics. The projects always begin with crystals. The goal of the structural biology project is to develop some kind of molecular picture, determine where are the atoms located within the molecular structure. And this is just one portion of the structure that I solved as part of my postdoctoral work, showing two strands of DNA, and they're coalescing or folding together to form a four-stranded structure where each of these layers is comprised of 4 guanosine bases, and then you can even see ions, probably potassium ions or sodium ions, going through the central axis of this molecule. But to obtain this molecular picture, we have to begin with some kind of crystalline sample. Inside of these crystals are these very complicated macromolecules. They're only showing here the non-hydrogen atoms. Here's the portion that we were looking at more closely previously, and at each of these vertices is some carbon, nitrogen, or oxygen atom that make up the structure of, in this case, 2 protein subunits that coalesce around a piece of single-stranded DNA. And this is only a fraction of one unit cell, and then these unit cells are repeated to form the whole macroscopic crystal. So to get to that point where you can grow the crystals, you need to prepare your macromolecules, and they have to. Very typically, we begin with crude extracts from bacteria that have obtained the information from making the protein through transformation with plasmid DNA. So these are the protein before purification, and after several steps, we obtain a sample where the protein is nearly homogeneous, or 99% pure. And you can also, people who are familiar with these type of SDS polyacrylamide gel electrophoresis results will appreciate that there's a lot of protein in this sample. We need approximately 10 or 20 milligrams of protein to begin exploring how to go from the aqueous phase into the crystalline phase. So,
I'm sorry.
Yeah.
How do you separate the proteins from the gel to rerun them? That's
Well,
what you do, right?
To go from here to here, there was a ammonium sulfate fractionation. So I add salt, and some proteins will precipitate at a low salt concentration, others require more salt. So there's a crude fractionation based on the interaction with ammonium sulfate. Then that sample is put over an ion exchange chromatography column, and So the proteins interact with the column at differential strengths based on their charge properties. And this is a DNA binding protein, so we used a negatively charged solid stationary phase, and the protein sticks to it in low salt, but comes out, goes into the mobile phase at some characteristic ionic strength. And then the final sort of polishing step was we took that fraction that was most enriched for the protein and put it over a size exclusion column that separates macromolecules based on size. So it takes about a week of work once you've got all the details worked out. This is again part of my postdoctoral work, and the lab that I joined had worked out many of these steps. And then these were actually purified here in Utah. I took those two subunits and I fused them together to make these engineered proteins. So I just borrowed the same methods, and they worked well in this case. There are a lot of bottlenecks. The protein may not be soluble when you make it in E. coli, and the protein may not be properly folded, or it may not. The search for getting crystals is sometimes really heartbreaking. I went to this one seminar, and it was a workshop for crystallographers, and you know the one speaker is talking about having made several, maybe 100 different versions of the DNA encoding the protein from different species, and testing them in different growth conditions and different purification schemes, and then finally testing 10,000 or more conditions for making the crystals, and eventually was able to, after years of hard labor, get crystals. And the next speaker said, well, we purified the protein and the crystals were obtained upon concentrating. I mean, just like one step with only one trial. So it's a very, there's a lot of luck involved.
How similar is the crystallized protein to one that's operational in?
Yeah, this is a very good question. The crystals of hemoglobin will bind oxygen, and there will be a structural transformation upon binding oxygen, as expected from allosteric properties of this molecule that transports oxygen from the lungs to working tissue. And so and there are other examples where you can actually, the enzyme, like for DNA polymerase, will actually catalyze the DNA polymerization reaction in crystalline form. So, crystals of small molecules are typically very dehydrated, so you'd expect a very, very different kind of environment. But these crystals, the unit cell is about half filled with protein and then half aqueous. So there's a lot of, there's still very important to keep the crystals hydrated, and in order for them to retain diffraction properties. And they often retain their biological properties, even though they're part of a crystal. So, and another argument is that
keep them hydrated.
You got to keep them hydrated, absolutely. And that actually was what Rosalind Franklin did well, that nobody else had thought to do, was to keep these DNA fibers hydrated. If you let them dry out, you don't get any interpretable diffraction pattern. And the same goes for these protein crystals. All right, so this is what you can see with a light microscope, and then at the microscopic level, the atomic level, there's a unit cell with a characteristic point group symmetry, characteristic dimensions. In this case, the unit cell belongs to a hexagonal space group, and you can see the morphology of the crystal matches that. So A and B are the unit cell axes are in this plane, and then the C axis runs in this direction. Here are some other examples of crystals that I've obtained in the lab. This is a complex of an inhibitor. It's a smaller protein, about 60 amino acids, in complex with the enzyme trypsin. This is a collaboration with David Goldenberg when I first got here to Utah. Lysozyme is like the gold standard. If you're getting discouraged and you can't make protein crystals, you can go buy some lysozyme and you make these beautiful crystals. Even my son Noah, he's now 22 years old. He was able to make these at a very young age. And they diffract really well. Okay, so here is the diffraction experiment. Once you have this biological material in a crystalline form, we shine X-rays through the crystals, And most of those X-rays go straight through the crystal. The X-rays interact with matter very weakly, and that's why we need crystals, is to amplify this weak interaction. And then there are diffracted rays that go that diverge from the direct beam, and we describe their one way to describe it is to what degree they diverge from the direct beam using this angle 2 theta. And here are some examples. Each of these little black flecks is caused by diffracted X-rays impinging on a detector screen and then getting recorded. And you can zoom in and then obtain the intensity of these diffracted rays. And each one of these diffracted rays has an associated index. We use the H, K, and L values and indexing them, indexing their diffracted rays has been automated through the computer advances. Okay, so this is time now for my demonstration. I didn't think to do this, but David Goldenberg told me about this, so I'm going to give it a try. I have here a laser pointer, and inside of these cells is a diffraction object. It's actually a grid that you would use for obtaining samples for cryo-electron microscopy. So it's just carbon filaments at, evenly spaced, and we'll see if I can get this to work under pressure. See, okay, so that's with
better than I see when I look through the curtains of a street light.
Right. Yeah, you could do this by squinting through a mosquito screen at a light, and so what we're seeing here is that the pinpoint light is now each one of these cells is acting as a point source, and you get constructive and a lot of destructive interference from those point sources, and you get this diffraction pattern. And there's a certain wavelength dependence to this. If you use a red laser or a green laser for the same grid spacing, the wavelength will determine to a degree the position of the spots. And also the spacing of the grid itself will change and be reflected in a reciprocal manner with the spacing of these diffracted spots. Bragg's law is one construct to explain why we get constructive interference, and you can think of the light being divided up into several parallel rays, and they are meeting up with a periodic structure defined by these Miller planes. Each set of planes is defined by its geometry with respect to the unit cell, a certain spacing between the planes, and then this ray here goes a shorter distance compared to this ray and compared to the parallel neighboring rays. And that distance is two of these A units, A being the spacing times the sine of theta. And theta is half of this observed 2 theta deflection from the direct beam. So you're putting all this together. If the wavelength of the light source is some integral number of wavelengths is equal to this two of these A units, then you'll get constructive interference. And everywhere else, you will get destructive interference. Okay, so this is what the equipment looks like in the biology department. We place our crystals here. To avoid damage from the X-rays, we keep the crystals frozen with a stream of evaporated nitrogen. The X-rays, the direct beam is very powerful, so we don't let that hit the sensitive detector. There's a direct beam stop that collects that direct beam right after it leaves the crystal. The source is a rotating copper anode. It's being bombarded with a array of electrons, and then the X-rays are the recoil from as electrons in the orbitals relax back to their inner shell positions. And then we move the crystal using a 4-axis goniometer, and then collect the diffracted rays with the use of a detector, a CCD detector in this case. And here are some additional examples of diffraction patterns obtained in the biology department. This is the crystals of BPTI and trypsin, and this is this DNA protein complex. It has a very the position of these spots is completely determined by the geometry of that experiment and the properties of the space group and the unit cell dimensions. But to collect the really good data, we travel to a synchrotron light source, and this is the one that I usually go to is located in Berkeley National Laboratory. It's called the Sibbles beamline. Dr. Scott Clausen facilitates getting me started on experiments at this site. And inside of this round building, there's a structure that looks like this. This what the photograph happens to be from the Swiss light source, so it's located somewhere else. These are all over the world. And inside, inside the ring, the so-called storage ring, is a stream of elementary particles, either protons or electrons, and they are held in position by carefully calibrated magnets. That at each of these sort of vertices, there's a strong bending magnet that changes the course of the elementary particle, and then from our understanding of conservation of momentum, there's going to be a large amount of energy recoiling every time you change the angular momentum of those particles. And they're traveling nearly at the speed of light around this ring. So you get a tremendous amount of energy tangential to the orbits. And it used to be just like a waste product of the experiments that were going on inside of the ring. But then people figured out this would be a fantastic source of X-rays. And the X-rays at home, they are, I'm limited to just one wavelength, but here the X-rays are, there's a whole spectrum of X-rays, and other portions of the electromagnetic spectrum are derived from. So each station sort of collects some sliver of that they really care about in infrared or ultraviolet or and then for these protein or macromolecular ended stations, the wavelength of interest is close to 1 angstroms in the X-ray regime. Here's this is like if you're at these places, they monitor the status of the ring all the time. Here there's 400 milliamps at 1.9 giga electron volts. That's in the storage ring. I don't use that energy; it's too powerful. But the light that comes to the end station is derived from this. You can see here there was some interruption at about 9 o'clock last night. They lost the ring. They had to put it back together. Here's what it looks like inside the hutch at 12.3.1. Everything is bigger. The detector is much bigger than the detector I have at home. The crystal sits here on the end of a goniometer. This goniometer only goes in one, there's only one axis for this goniometer, but it's enough. The detector is so big that we can collect all the data from here. Here's a comparison of diffraction pattern obtained at the synchrotron and at home. I mean, the detector is much larger, so the area able to cover at home is just a fraction of what I can capture at the synchrotron. This piece of equipment would cost just way too much for me to attempt purchasing here in Utah, but I get to use it essentially for free at the National Laboratory. And the other thing is that the X-ray source is so much brighter at the synchrotron that a diffraction pattern like this would probably take about 5 minutes to collect. And here at the synchrotron, I usually collect each of these photographs takes about 1 or 2 seconds to obtain. And you don't just capture one pattern, you capture hundreds of these at different geometries where you're moving the crystal through uniform angle steps. And so that is, it ends up being a 20-minute data collection in place of a 3-day collection. And the other thing is that at the synchrotron, you can capture these very weak diffracted rays that won't ever show up, no matter how long you wait for them at the home source because of signal to noise. And so there's a real advantage to going to the synchrotron. So here's a movie that shows the
How do you control the X-ray wavelength?
There are optics that and very fine-tuned motors that sort of filter out the unwanted and enable you to tune. And it's difficult to get X-rays to reflect off of things, but they have these polished surfaces,
Yeah.
very fine-tuned motors. So here, the crystal is making about 0.5 degree steps for each one of these photographs, and each different geometric position you bring into a position where certain subset of all the diffracted rays will satisfy Bragg's law, and the job after collecting all these photographs is to now integrate the intensity at each of these positions, and also index so that you know exactly the Miller planes and the spacings for and orientation with respect to the crystal for each of these diffracted. It's hard to see the individual rays in that view, so this movie shows a zoomed-in view, and each one of these spots is the result of one diffracted ray. And the unit cell, I think I said this before, but the unit cell and space group determines where the diffracted rays will fall on the detector surface. But the difference in intensity from one diffracted ray to another, that's the source of information for the structure, the substructure of the crystal, or the structure of your macromolecule. So I'm just going to, in the interest of time, we have to keep this at a fairly conceptual level. We could spend weeks describing all of these things. We've seen how Bragg's law explains why we get diffracted rays at certain positions on the detector. And this is going from real space to reciprocal space. The large spacing here reflects or is determined by the smaller unit cell, this 94 angstrom unit cell dimension. This very close spacing here between this train of spots, that's determined by the longer 426 angstrom unit cell dimension, the C-axis. Now, to go from reciprocal space, this information, and convert it into a form usable in real space, we calculate the electron density map from data collected like this, and we use the fast Fourier transform, so the Fourier transform of reciprocal space to go back into real space. And here's the
Mechanically read the intensities?
yes, it's all automated. I
hope so.
This was the ability to determine the structure of myoglobin and then hemoglobin was absolutely dependent on the proximity of the first computing machines. There was a room filled with women who were looking at the photograph films and scoring each of these
They were called computers.
They were I think they're right. Now I feel very lucky. The indexing, the attention to symmetry that my advisor had to pay in order to figure out where the spots were located and to be able to name them, that's all taken care of using computer software. And so, it's you can almost think of it as like a black box, but we're trying to give it some foundation for what's going on inside of those black boxes here. So, calculating electron density, we use this. We take this Fourier sum over the volume of the unit cell. We have indexed HK and L reflections. We can record the amplitude of the diffracted rays, and that's what the absolute value of this F sub HKL represents. And then there's the convolution or the dot product of the position in the unit cell X, Y, and Z with these HKL values. And one of the really important pieces of information that we cannot measure directly is this phase associated with each of the diffracted rays. Yeah.
Oh, you mentioned the two scales. Were you speaking of a sort of a banded structure, and then is being the structure of a hemoglobin molecule in this case, or and then the fine spacing is the way they kind of stack themselves together. Oh.
Yeah, it's this spacing here. This is like just like you can think of it like shoe boxes, and there's a short length to the shoe box, And that determines how far apart things are here. You can actually, with software, you can draw a line that transects several of these, and it will tell you what the unit cell is in that for that direction. And then you draw another transect here, and you'll get like these spikes, and the distance between them determines the longer one. It's the idea that in real space and reciprocal space are inversely related to each other. So things that are far apart in real space end up very close together in reciprocal space.
If they didn't stack in an organized way, you can still try to...
Okay, so it has to be crystalline, yes. If it's amorphous, if you don't have a regular repeating structure, then you won't get this constructive interference that it won't diffract. And the solution there is to switch to some other source of radiation that interacts more strongly. And that...
They look bright enough. I mean, the bands are so clear and defined.
Yeah.
It seems like they'd still be resolvable.
Yeah, but that's from a diffracting crystal. There are plenty of crystals that don't diffract. So that you would not see any spots if you didn't have this organized repeating structure to the crystal. Okay, so calculate electron density with the Fourier transform. Now you can build into this. The electron density map is a four-dimensional kind of a structure. At each x, y, z position, there's some prediction of electrons being located there, and atoms are located where electrons are. And these are all contoured at an equal electron level, and then you can adjust that contour level. Yeah.
So you were starting to say what you did about the phase.
Yeah, well, we're not going to go very deep into that. This is where most of the information for this structure actually is in this term here, which we can't measure directly. And as we're going to see later, it becomes very important to know what this phase is and to get the structure. We solve the problem in a number of different ways. If the substructure is small enough, you can just guess and use heuristic methods, and that's what's applied for small molecule crystallography. Up to 600 or even 1,000 atoms. That's a practical approach. For proteins with 5,000 non-hydrogen atoms, it becomes difficult to do. And what you can do is, if you know the structure of a similar protein, a homologous protein, you can use molecular replacement. It may not be in the same space group, but you can make guesses as to its rotation. You can separate rotational search from the translational search and obtain a satisfactory guess that then gives you approximate phases to calculate initial electron density maps, and then you iteratively improve the structure and get better maps. And then the original method, worked out by Perutz and applied by Kendrew first, was this thing called multiple isomorphous replacement, where you add heavy atoms to the crystals, you soak them in, and they bind at specific locations within the crystal, and sort of and because they're heavy atoms, they interact with the X-rays so much more strongly than do the carbons and nitrogen and oxygens. And you solve the substructure for the heavy atoms, and then by comparing the intensities with and without the heavy atoms, you can make good guesses as to the phase for each of the reflections.
The phase is just the complex phase of F, right?
Yeah, exactly. It's a complex component, and then the more modern approach is that you grow your cultures in a species of bacteria that does not make methionine, and you supplement the medium with selenomethionine. And now, at each place where normally you'd have a sulfur atom, you now have a selenium atom. And that has, at a synchrotron, you can tune the wavelength to match the anomalous scattering edge for selenium, or whatever atom with anomalous scattering properties in your sample. But selenium is a convenient choice, and there get phases directly from one crystal without having to compare diffraction patterns. Yeah, it's a tricky problem. Okay, so we build a molecular model. This is positions of the atoms. And then once we have the model, we can go back using the inverse fast Fourier transform and calculate what the properties are of the diffracted rays. And in this case, we're summing over all atoms. Again, each atom has a defined location within the unit cell. And these so-called scattering factors for each atom are known and recorded for X-rays and other radiation sources. And there's a wavelength dependence, and so you have a lookup table, and you can calculate. And this is important so that you can check your model against your data by comparing the amplitude of this calculated F calculated from the inverse Fourier transform. Compare it with what you can measure, the amplitude of the observed diffracted ray, and this is the metric that we call the R value, and that sort of that guides the refinement, the repositioning of the atoms, either in an automated computer-driven mode or through manual inspection of electron density maps, the X-ray crystallographers adjusting position of groups within the structure. Here's the whole thing mapped out in a we call it the refinement cycle. Beginning with your data, some initial guess for phases, you can compute the electron density map. The electron density map is then used to build the model or adjust the model. If things are going well, your electron density maps in subsequent cycles will tell you that there's pieces of the model that are not in the right position, and then you can iteratively go through the cycle, always checking your work by calculating the inverse fast Fourier transform and comparing the absolute value of your f's with the ones that we're calculating. Things don't always go well. There's two possible ways that the refinement cycle can you can either converge to a true, more accurate structure, or you can just introduce more errors in the structure to compensate for the errors that you already have. And so people in the 80s worked out, especially Axel Brunner, worked out the idea of you reserve some portion of your data, 5% of your data, 10% of your data, which the computer never gets to see, and it's put into this test set. You compute the R value for the working set, and that sort of sets up gradients for moving the atoms in certain directions during refinement and also during model adjustment. And then you compare, you monitor how the R values are behaving for the working set, but you also pay close attention to what's happening to the R free value. If your model bias is completely determining this, then R free will go up. And if you're actually getting close to the true structure, then the R free value will go down in parallel with the R calculated for the working reflections.
I don't understand why you keep the secrecy from the computer.
Well, it's so that the idea is that if the computer got to see those reserved reflections, it would kind of contaminate or bias we're worried about, because the phases are calculated from the structural model, we're worried that you're just going to get back something that's self-reinforcing or self-assuring. So you fool yourself that there's classic examples where people have traced the polypeptide chain of the protein exactly in reverse, and it's completely impossible. And to guard against that possibility, we keep 5% of the data, 10% of the data secret.
It saves you from that.
It saves us from this trap. Yeah, exactly. If you have a lot of data, you don't need to continue to. You can keep just 2% of it secret or 1% of the data. So as your resolution limit improves, you're collecting higher quality data. The data to parameter ratio is satisfactory, so you're unlikely to fall into this trap. But if you're at a modest resolution limit, like 2.5 angstroms, 3.5 angstroms, then you have just barely enough data to determine all of those parameters, all of the XYZ positions and their temperature factors. So, okay, we're coming to the end of the introduction. This has gone a little bit longer than I wanted it to. I hope if none of that made sense, or if you were asleep, I can recommend this very short 2.5 minute video put out by Oxford Sparks. It's really cute, and at least for crystallographers, it is really funny. I don't have time to show it. because we're going to talk about research. All right. This is a project that I started with Sheila David while she was still here at the University of Utah in the chemistry department, and she's now moved to UC Davis. And we have just recently been able to publish the work, so we're really excited that the structural biology has been able to make significant advances. Now, the biology of why this system is important has to do with DNA damage. Here's double-stranded DNA showing a GC base pair. G is the one nucleotide, the one base that's has the lowest redox potential, most susceptible to oxidation. And in the OG form, it can base pair with either A or C. So under rounds of replication, you're at risk of forming these base pairs where adenine is base paired with this modified form of G. And then one further replication step, you would end up with a mutation. So the evolution has come up with a way to cope with this and to repair the DNA before getting to the mutated form. To show exactly why you can fit adenine across from guanine in the active site of DNA polymerase, here's the active site of DNA polymerase showing an OG base paired with cytosine. This is a normal Watson-Crick type of interaction and would not lead to mutation. But here's that same modified G. The extra oxygen is right here. And now it's flipped over and presenting not the Watson-Crick face, but the Hookstein face, where it can hydrogen bond with adenine. So the addition of oxygen here changes this nitrogen from a hydrogen bond acceptor to a hydrogen bond donor, and that now makes it possible for these two atoms to mimic the properties of T and template the insertion of adenine. Here's this movie just makes it painfully obvious what's going on. We have conversion across this N-glycosyl bond, and this base is therefore dangerous because it can template for either the correct nucleotide in the DNA, C, or the wrong nucleotide, adenine. There are multiple layers of multiple DNA repair enzymes that cope with this potential source for mutations. One of them is MUT-M that removes the OG, the initial offending chemical lesion, and then allows for repair at this site. But these OG-A base pairs accumulate frequently enough that there's a backup system to prevent mutation. MUT-Y intercepts this base pair and removes the adenine from before it can before a round of replication. And deficiencies or changes or variations in this enzyme in the human population lead to a predisposition for developing colon cancer later in life. And those were detected by this GC to TA mutation pattern. Normally, you don't expect a G to become a T or a C to become an A. It's the more rare kind of mistake for DNA polymerase to make. But in these patients with colon cancer, they had a lot of these GC to TA transversions. And so people studying MUT-Y deduced that there must be something different about the MUT-Y enzyme. And so instead of the typical like BRCA1 where you have a tumor suppressor gene or an oncogene which gets inherited, what you have is a slightly underperforming MUT-Y enzyme that allows somatic mutations to accumulate, and eventually you hit one of those tumor suppressors or oncogenes. So in Sheila's lab, Valerie, at the time was a graduate student, asked the question, what's the catalytic mechanism of this enzyme? And to describe the catalytic mechanism, she set out in partnership with me to obtain a crystal structure of this enzyme, MUT-Y, in complex, not with the substrate, but with the transition state, a transition state mimic. And this is something that Sheila David, organic chemist, can synthesize and then incorporate into DNA. So the DNA molecules in these crystals, they contain regular Watson-Crick base pairs, but they also contain this OG paired, not with adenine, but with this transition state mimic. And the transition state is expected to be highly reactive and only exist for a few bond vibrations. And so in order to buy the time needed to make crystals and analyze the crystals, we have to use a stable mimic form. And so it's expected to have the similar charge properties similar shape properties, but it's different from the transition state. It's not reactive, and we don't expect reaction in the crystal form. And this is now an overview of the protein structure. We're tracing the course of the polypeptide chain with this ribbon. The DNA is texturized. Base pairs are these yellow and gray things. These are 2 amino acids that are inserted into the stack of bases, and that causes a distortion in the structure of the DNA to pull the substrate into the active site. And what we're seeing here is now sort of a view of an intermediate expected during the course of adenine removal, where we have this one-end transition state mimic interacting with catalytic residues at the active site. And there are a large number of statistics that sort of back up that we did the job carefully. I'll just call attention to this one here. This is the resolution limit. This is the minimum D spacing for those Miller planes, and that then gives you confidence that you can determine at about 2 angstroms. You can your coordinate error is about 0.2 angstroms. So you're confident about hydrogen bonds and other types of molecular interactions. The R values, this is 24%, seems really high, but this is acceptable for publication. The lower limits for R values, even if very good data, is about 15% for this type of work. For small molecules, you need an R value of about 5%, but for these large molecules with this is very typical parameter values for a good structure. Okay, so we're just going to we're going to look at the structure, just some of the highlights. We're looking here at the active site, and I'm trying to describe how things get in and out of the active site. And here we have the one-end transition state mimic, and then we have a nucleophile, a water molecule we expect a water molecule to intercept the transition state and finish the reaction after adenine removal. So we're looking at the location of water molecules. Here is now the active site shown with electron density clothing the residues, and here's the electron density for this water molecule that we've labeled C in the publication. This aspartate D144 is absolutely essential for catalysis, and we knew that before we did our work. This tyrosine 126 was not thought to be a catalytic residue, but its close proximity to the transition state alerted us to the possibility that it is involved in stabilizing the transition state. So Valerie went and made mutations replacing this tyrosine with alanine or with phenylalanine and with serine, and then showed that the enzyme is much slower. Not completely inactive, but it's certainly an important residue for it. So we're getting insights as to what is the catalytic mechanism and what stabilizes the transition state from these structural views. Here, this is now a synthesis of a lot of information that I'm not going to show. People had already worked out the beginning part of the reaction, how this bond between the DNA strand and this nitrogen is weakened through acid catalysis. And then what we propose now is that this water molecule must be attacking from the same face from which adenine is departing. And that was new and not easy for people reviewing our work to accept. So we had to provide a lot of corroborating evidence, including determining the stereochemistry of the product. The time needed for adenine to leave and for water to come in is probably on the millisecond or microsecond time scale, and that transition state is expected to be very unstable during that time. So we are invoking or inferring that this aspartate, in addition to providing electrostatic interactions that stabilize the transition state, also forms a covalent bond. So we're predicting that there's a covalent DNA enzyme intermediate, so using covalent catalysis. Okay, this is a chalkboard drawing from the course I teach, biochemistry, and hydrogen bonds are easy to draw, and and difficult. It's much more difficult to appreciate or to show the steric properties. But much of what we much of the inferences that we obtained for the mechanism were included thinking about steric repulsive forces. And so I'm exploring ways to show these steric complementarity and when do things bump into each other. We would like to know where is the adenine going after it's been detached from the DNA. And Sheila has been able to construct additional transition state mimics where there's a moiety that sort of pulls the large group further away from the departing carbon atom. And I'm modeling here in the active site what are the possibilities defined by constraints imposed by steric, what would be steric clashes. So we see here that tyrosine and these are the catalytic residues, and this molecule is meant to mimic a portion of the adenine as it's departing from the transition state.
What's steric?
Steric it has to do with like shape and like it's this idea that atoms can't occupy the same space at the same time. Here now she's able to embellish this group and has added a much larger group, and I'm trying to map out where in the active site such a large group would end up if it was trying to leave the active site, and that led to the realization that there's a second access portal that adenine may be able to be departing from the active site through here, even as water molecules coming in from that first access portal.
So, a couple questions. One is, it seems like you have a lot of space around. Can you replace that tyrosine instead of alanine scanning, alanine mutation? Can you put a tryptophan in there?
Oh, that's a good idea. No, we have not tried to do that. I would predict
Push it the other way.
Yeah, push it the other way. I predict it would be highly impaired. But that's a really good test of the
And
idea.
the other question is, what would you expect? This is not. What would you expect for heavy water?
Okay, people have done kinetic isotope effects and determined, and based on those, that was the beginning part of the mechanism. The adenine has to dissociate first, and then water is added at a second step. So it also informed like glutamate 43 is likely to be that acid catalyst in that situation. So yeah, people do use heavy water to determine the rate of the reaction under those conditions. Okay. All right. So we're just going to look quickly at this final system. This is where my head has been for the last few days, trying to understand how these carotenoid molecules fit into proteins found in the human eye and seem to be protective against age-related macular degeneration. So in the macula lutea, here's a portion of the retina. These molecules are concentrated, and the biochemistry underlying how this spot develops and how it protects the eye is of great interest for Paul Bernstein, my collaborator, and his postdoc, Benjing Li. We don't make these molecules ourselves. We have to get them from our diet, and so this is why we eat brightly colored fruits and vegetables. And egg yolks is another great source of these molecules. We do not get this molecule from our diet. There's some metabolic conversion of lutein to meso-zeaxanthin, and so one area of exploration is exactly how does that happen. In the initial project, we made crystals of this protein called STAR-D3 that was discovered by binging to bind to lutein in the human eye. And Evan George was an undergraduate in the lab, and he was able to purify the protein and make these crystals. And we were able to get this is a sample of lutein in a dry powdered form. We were able to get a colored complex to form in the crystals, but unfortunately these crystals, they do diffract, but they don't show any evidence that the molecule is actually bound in the active site. So just to keep expectations low. Here's diffraction pattern from these crystals that Evan grew, and then this is the crystal structure that we determined, highlighting areas where things can get in and out of the central core of the protein. There are these two portals. Portal 1 is much larger than portal 2. It's guarded by this loop, which shows evidence of flexibility. And then I've highlighted with in red and the surface representation water molecules that are filling up this channel in this middle of the protein. Most proteins, the typical protein, like if you slice hemoglobin through the middle or myoglobin through the middle or lysozyme through the middle, you're not going to see a large empty tunnel like this. So this is a special feature of this class of proteins, and it's probably designed to bind to hydrophobic molecules. Here's another example. This structure I did not determine, but I obtained the structure with use of already determined structure of the enzyme from cows, and then I threaded the sequence of chicken protein to obtain a homology model. So we're in the realm of model building, not experimentally determining the structures. And the question is, can we fit lutein in somewhere into this structure? And I'm using rigid body molecular docking in a very crude way. I've broken the problem down into 4 steps. I find the empty spaces. In this case, it's guided by an inhibitor of the enzymes, and you can see through this cutaway slice view that there's a lot of empty space above the active site that's located here. I define the structure of the ligand by examining examples where I see lutein in complex with proteins, and those are the chlorophyll binding proteins that assemble with the photosystems. And so there's about 40 reliable structure. It's not flat. It has curvature, and so to make sure that I'm working with a ligand structure that's reflects the authentic molecule, I borrowed those 40 structures and I'm to define the structure of the ligand. And then it's just a puzzle building, trying to fit those ligand structures into this empty space, and I'm scoring by steric clashes and hydrogen bonds. So how many conflicts are encountered, and how many favorable interactions. Here's like I've placed all of these, I call them guidepost atoms. They're just water molecules to fill up the empty spaces. Some of them are derived directly from the structure of the inhibitor as found in experimentally determined structure, and others are I'm just guessing and making sure that there's no collision when I place the water molecules there. And then here, see that this is like the view after I painstakingly positioned all these pseudo guidepost atoms. I could sort of squint and see the structure of a ring, and then I'm using least squares position, picking a subset of these guidepost atoms to guide the positioning of the lutein molecule. And then this is now at the other end of the molecule. I look at how it fits not only in that region, which is now buried inside of the structure, but also how the molecule emerges from, make sure there's a direct route to get in and out of the active site. And then you can see here, it's not a perfect fit. There are places where there's steric conflict, and I count those as, and I also look for what would be hydrogen bond interactions, which would be favorable, and computationally sort and score all the possibilities. Here's the outcome of this sorting and scoring for docking lutein in STAR-D3. You can see there's a lot of clashes. Many times, the way I'm doing it, things don't work. By comparison, these are the lutein molecules as found in the chlorophyll binding proteins. They make very few clashes, and they make several hydrogen bonds. Here, the best winner that I have makes 14 clashes and only 2 hydrogen bonds. But still, I think it's a starting point to to make inferences about the complex that hopefully we can replace with experimentally determined structure in the future. Here are some close-up views of what lutein looks like in complex with STAR-D3. Here's one of the hydrogen bonds, and then the second hydrogen bond. And what this shows is the number of clashes for all of the outcomes. And the distribution shows that this epsilon ionone ring with a chiral carbon here, an sp3 hybridization, is more likely to have fewer score better in these tests than the beta ring, which usually has more clashes, suggesting that there's the shape of this cavity is designed to accept or select for with specificity the one end of the molecule as opposed to the other end of the molecule. Okay, we're done. These are my acknowledgement slides again. You've seen all these people already. I want to thank Valerie and Sheila and Scott for the MuteY project, Paul Bernstein, Binging Lee, and Evan for the carotenoid-binding project. NSF funding, Sheila and I just applied for and got funding to continue our work with MuteY, and then Paul Bernstein has a grant with the NIH. Yeah, thank you. Questions?
So in the docking, do you also factor in the kinetic limitations of how it would thread itself into that position?
No, I have not done any simulations to see how it would get in there. I'm essentially just putting it in there by hand, and then what I'm looking for is a source of variation in position and shape that would still retain realistic structure for the ligand. I don't allow the protein to relax. I mean, there's a lot more sophisticated modeling that could be done, but as a starting point, I call this, it's just really coarse grain. Does it fit at all, or if it does fit, is there any evidence that there's the shape or the asymmetry in the ligand is reflected in asymmetry found in the protein structure? So that's what I hope for. Yeah.
We hear about proteins folding and unfolding all the time, like that's an important thing. Can you catch them and crystallize them in different configurations?
Certainly, the protein, some proteins adopt alternate conformations, and we see that at many different scales. We'll see amino acid residues in two different positions. But the thing that you're really interested in is this intermediate in the folding pathway. By its very nature, you would not be able to crystallize it because it's so dynamic.
One end or the other.
Well, the unfolded form would be very hard to crystallize. So David Goldenberg works on modeling and many people work on what the behavior of the unfolded state is based on either computational modeling or you can use a small angle scattering in the where you just use a liquid sample, not crystals, and that's another method that can give you some structural information. But for this you need a structured form to make crystals.
Why do you see the reciprocal lattice and not the actual thing first? What are you measuring again?
Okay, so yeah, it has to do with the fact that you're passing light through this repeating pattern. If you were to pass the light onto one copy of the molecule, and then you could capture the image directly, right? And that's what you do with electron microscopy.
Right.
And up until recently, the resolution limit for electron microscopy was about 20 angstroms. There have been improvements such that now, in some cases, cryo-electron microscopy, where you're looking at single particles, one at a time, and actually capturing and reconstructing their images, you can get to 2.5, 2.2 angstroms. So close to what, there's now an overlap between the resolution for this work that I'm showing you and and more direct imaging methods. But yeah, the reason why you're in reciprocal space is because the light's not hitting one particle, it's hitting multiple repeating units all at the same time, and therefore you end up in reciprocal space.
Okay.
That diffraction pattern is the Fourier synthesis of your crystal. So.
Are the people using neutrons?
Yeah, neutron diffraction. You need with X-rays. The scattering factor for hydrogen is very small, so you don't normally see hydrogens except at very high resolution. 0.6, 0.7 angstrom resolution. But neutrons, they interact strongly with hydrogens, and so you see those. It's like the opposite of what with X-rays. But you need a very large sample. You need crystals much bigger than the ones I've shown you, and you need specialized neutron source. And the data collection, I understand, takes a very long time, even at those powerful neutron sources.
Sometimes people take collections of crystallites or crystals, small crystals, and orient them and paste them down and do scattering from them.
Yeah, it sounds amazing.
Well, what's crazy?
Yeah, for my work, it has to be a single crystal, and there is this stage called
that's right.
polycrystallinity, where if you have 2 or 3 neighboring crystals, they will make overlapping and interfering diffraction patterns,
Right.
and then there's an even more insidious kind of thing where the space group symmetry axis can change direction within what morphologically looks like a single crystal, and you get so-called marihedral twinning, and there are computational methods to deal with those, but you need good data in order to cope with it. All right, thanks so much. Yeah. Everything out of my...
That is irritating.